Plant cells are eukaryotic cells that differ in several key aspects from the cells of other eukaryotic organisms. Their distinctive features include a large central vacuole, a water-filled volume enclosed by a membrane known as the tonoplast that maintains the cell's turgor, controls movement of molecules between the cytosol and sap, stores useful material and digests waste proteins and organelles. A cell wall composed of cellulose and hemicelluloses, pectin and in many cases lignin, is secreted by the protoplast on the outside of the cell membrane. This contrasts with the cell walls of fungi, which are made of chitin, and of bacteria, which are made of peptidoglycan. Cell walls perform many essential functions. They provide shape to form the tissue and organs of the plant, and play an important role in intercellular communication and plant-microbe interactions. Specialized cell-to-cell communication pathways known as plasmodesmata pores in the primary cell wall through which the plasmalemma and endoplasmic reticulum of adjacent cells are continuous. Plastids, the most notable being the chloroplast, which contains chlorophyll, a green-colored pigment that absorbs sunlight and allows the plant to make its own food in the process known as photosynthesis. Other types of plastids are the amyloplasts, specialized for starch storage, elaioplasts, specialized for fat storage, and chromoplasts, specialized for synthesis and storage of pigments, as in mitochondria, which have a genome encoding 37 genes. Plastids have their own genomes of about 100 to 120 unique genes and, it is presumed, arose as prokaryotic endosymbionts living in the cells of an early eukaryotic ancestor of the land, plants and algae. Cell division by construction of a phragmoplast as a template for building a cell plate late in cytokinesis is characteristic of land plants and a few groups of algae, notably the charophytes and the chlorophyte order Trentepoliales. The motile, free-swimming sperm of bryophytes and pteridophytes, cycads and ginkgo are the only cells of land plants to have flagella similar to those in animal cells. But the conifers and flowering plants do not have motile sperm and lack both flagella and centrioles. Cell types. Parenchyma cells are living cells that have functions ranging from storage and support to photosynthesis and phloem loading. Transfer cells. Apart from the xylem and phloem in the vascular bundles, leaves are composed mainly of parenchyma cells. Some parenchyma cells, as in the epidermis, is specialized for light penetration and focusing or regulation of gas exchange, but others are among the least specialized cells in plant tissue, and may remain totipotent, capable of dividing to produce new populations of undifferentiated cells. Throughout their lives, parenchyma cells have thin, permeable primary walls enabling the transport of small molecules between them, and their cytoplasm is responsible for a wide range of biochemical functions such as nectar secretion, or the manufacture of secondary products that discourage herbivory. Parenchyma cells that contain many chloroplasts and are concerned primarily with photosynthesis are called chlorenchyma cells. Others, such as the majority of the parenchyma cells in potato tubers and the seed cotyledons of legumes, have a storage function. Collenchyma cells are alive at maturity and have only a primary wall. These cells mature from meristem derivatives that initially resemble parenchyma, but differences quickly become apparent. Plastids do not develop, and the secretory apparatus, in Golgi, proliferates to secrete additional primary wall. The wall is most commonly thickest at the corners, where 3 or more cells come in contact, and thinnest where only 2 cells come in contact, though other arrangements of the wall thickening are possible. Pectin and hemicellulose are the dominant constituents of collenchyma cell walls of dicotyledon angiosperms. which may contain as little as 20% of cellulose in petacytes. Collenchyma cells are typically quite elongated, and may divide transversely to give a septate appearance. The role of this cell type is to support the plant in axis still growing in length, and to confer flexibility and tensile strength on tissues. The primary wall lacks lignin that would make it tough and rigid, so this cell type provides what could be called plastic support that can hold a young stem or petiole into the air, but in cells that can be stretched as the cells around them elongate. Stretchable support without elastic snapback is a good way to describe what collenchyma does. Parts of the strings in celery are collenchyma. Sclerenchyma cells, from the Greek skleros, hard, are hard and tough cells with a function in mechanical support. They are of two broad types: scleriads are stone cells and fibers. The cells develop an extensive secondary cell wall that is laid down on the inside of the primary cell wall. The secondary wall is impregnated with lignin, making it hard and impermeable to water. Thus, these cells cannot survive for long, as they cannot exchange sufficient material to maintain active metabolism. Sclerenchyma cells are typically dead at functional maturity, and the cytoplasm is missing, leaving an empty central cavity. Functions for scleriad cells, hard cells that give leaves of fruits a gritty texture, include discouraging herbivory by damaging digestive passages in small insect larval stages, and physical protection. A solid tissue of hard scleriad cells form the pit wall in a peach and many other fruits. Functions of fibers include provision of load-bearing support and tensile strength to the leaves and stems of herbaceous plants. Sclerenchyma fibers are not involved in conduction, either of water and nutrients, as in the xylem, or of carbon compounds, as in the phloem, but it is likely that they may have evolved as modifications of xylem and phloem initials in early land plants. Tissue types. The major classes of cells differentiate from undifferentiated meristematic cells, analogous to the stem cells of animals, to form the tissue structures of roots, stems, leaves, flowers, and reproductive structures. Xylem cells are elongated cells with lignified secondary thickening of the cell walls. Xylem cells are specialized for conduction of water, and first appeared in plants during their transition to land in the Silurian period more than 425 million years ago. See Cooksonia. The possession of xylem defines the vascular plants are tracheophytes. Xylem tracheads are pointed, elongated xylem cells, the simplest of which have continuous primary cell walls and lignified secondary wall thickenings, in the form of rings, hoops, or reticulate networks. More complex tracheads with valve-like perforations called bordered pits characterize the gymnosperms. The ferns and other pteridophytes and the gymnosperms have only xylem tracheads, while the angiosperms also have xylem vessels. Vessel members are hollow xylem cells without end walls that are aligned end-to-end so as to form long continuous tubes. The bryophytes lack true xylem cells, but their sporophytes have a water-conducting tissue known as the hydrome that is composed of elongated cells of simpler construction. Phloem is a specialized tissue for food transport in higher plants. Phloem cells mainly transport sucrose along pressure gradients generated by osmosis. This phenomenon is called translocation. Phloem consists of two cell types: the sieve tubes and the intimately associated companion cells. The sieve tube elements lack nuclei and ribosomes, and their metabolism and functions are regulated by the adjacent nucleate companion cells. Sieve tubes are joined end-to-end with perforate end plates between known as sieve plates, which allow transport of photosynthate between the sieve elements. The companion cells, connected to the sieve tubes via plasmodesmata, are responsible for loading the phloem with sugars. The bryophytes lack phloem, but moss sporophytes have a simpler tissue with analogous function known as the leptomy. Plant epidermal cells are specialized parenchyma cells covering the external surfaces of leaves, stems and roots. The epidermal cells of aerial organs arise from the superficial layer of cells known as the tunica, L1 and L2 layers, that covers the plant shoot apex, whereas the cortex and vascular tissues arise from innermost layer of the shoot apex known as the corpus, L3 layer. The epidermis of roots originates from the layer of cells immediately beneath the root cap. The epidermis of all aerial organs, but not roots, is covered with a cuticle made of the polyester chitin and/or the hydrocarbon polymer chitin with a superficial layer of epicuticular waxes. The epidermal cells of the primary shoot are thought to be the only plant cells with the biochemical capacity to synthesize chitin. Several cell types may be present in the epidermis. Notable among these are the stomatal guard cells, glandular and clothing hairs or trichomes, and the root hairs of primary roots. In the shoot epidermis of most plants, only the guard cells have chloroplasts. Chloroplasts contain the green pigment chlorophyll which is needed for photosynthesis. Organelles